I'm Katerina Vrisa. I am a postdoctoral researcher at the Center of Nanoscale Materials in Argonne. And today I'm going to give a kind of different talk than the previous ones that were mostly focusing on training and designing machine learning models. So this time I'm going to present some tools that have been developed from our group here to extract data from literature. And I also some examples how we can use this data that we can extract and why they are useful, especially currently with the rise of multimodal data and foundational models. So a few things about me. So my main areas of expertise is applied AI machine learning, basically for materials. So mostly for molecular crystals or molecules or polymers. I'm also doing some DFT simulations for metal problematic hydrocarbons or molecular co-crystals. And I'm working at the autonomous lab at the CNM, where we are trying to design robotic platforms and actually use AI or simulations to drive the autonomous experimentation. So and currently we're focusing on synthesizing polymer electronics. So a tutorial overview. So in the GitHub repository that I have shared, I have some code examples that I want to run to showcase how the journal scrapping can be performed by a user very simply, and then also how we can use the data that we're extracting. So it's actually, I categorize them, okay, how we extract them and how we use them. So how we extract them is either we can directly extract from a journal. So for instance, given Nature, RSC, or Wiley. So we define the type of the journal we want to scrape from, and also some keywords. So what we're searching for. And it's actually connecting with the database of the journal, and then it's scraping all the related open source papers from there. And I also recently have added like a second functionality, which is an HTML scraper. So if you have a list of papers that you want to scrape particularly from them, then you can upload them to a folder and then call the HTML scraper to extract all the images and captions of all the journal data. And then the second part, I'm going to show, okay, how we can perform named entity recognition. So how we can identify some entities from the captions we extract. I also showcase how we can use CLIP model, which is a model that recently developed by OpenAI and can handle image and text. It is very used for queries. So by given an image, we can identify a similar image from our database or find a description through the text. And also at the end, I'll present, if time allows, like Plotospect, which is like a tool developed here that can handle like spectroscopy data, so that we can digitalize any spectroscopy data images. So first of all, why do we need multimodal datasets? So with multimodal, I mean mostly like text and images. So currently, there is a big rise in foundational models. So this is kind of the models that can be trained with different with data from different type formats. So for instance, with speeds, image, signals, text. So a model can use all this type of information, and then we can fine tune it and repurpose them for some specific tasks. For instance, it can answer questions, it can do sentiment analysis, image captioning, or object recognition. So it can be very generalizable and go beyond, give predictions for beyond the data types that it has been trained on. Very recently, many material researchers, they also want to apply this technology for material science. So how a foundational model for material science would look like is that we can train the model with all the type of information we have for our materials. So from small strings, from the molecules to the absorption spectra, or to maybe crystal structure information, or some synthetic recipes that we can make this molecule. So any type of information relevant to our materials. And so a foundational model can be more generalizable. So because some materials types might have quite similar properties, although belong to different categories. So by having a general purpose network and then fine-tuning it for downstream tasks can be very beneficial. But to do that, to train a foundational model, we need like data from many data types. And this is actually why we developed EXSCLAIM, which is a tool that can extract information from literature for any type of material we're interested in. So the current challenge when we want to scrape scientific literature is that is unstructured data. So and lots of the information also is not on the text or by it's mostly on the images. So the important information for our materials. And for instance, especially if you work in fields like microscopy, images are the most important part of the papers. And we want to be able to get them and analyze them. So this is how the EXSCLAIM workflow looks like. So EXSCLAIM means extraction, separation, and caption-based language annotation of images from a scientific literature. And this is kind of a high-level overview how EXSCLAIM works. So we start from a query. So we can give the name of the folder we want to save the tools we extracted. We also identify the journal family. So if we want Nature or ERASI, for instance, and then we show our queries. So what we are interested to extract. So palladium nanoparticles, for instance. So after sending this query to EXSCLAIM, then it goes through the journal database and then using Python requests to scrape all the HTML files from related to our research query. After that, we use caption distribution. So that means for every full caption that describes the image, we take the caption and then split it to sub-captions to describe the sub-figures. Then, based on the captions, we also have a vision model that is doing the figure separation. So from the whole, because usually in the journals, all the images are all together forming one big image. So, and we want ideally to separate them to the smaller subfigures. But then at the end, what is outputted is like an EXSCLAIM JSON that has all the information. So it has the title of the paper we scraped from, the URL, the full caption, also classification of the image, if it's microscopy, or if it's like absorption spectrum, the geometry, which is kind of the bounding box. So from the large image, which is the bounding box that describes the subfigure, and also the subcaption of sub-image. So I'll show some examples so that everybody can understand how this process works. And also, we try to integrate some language models with the EXSCLAIM workflow. So, for instance, we can also identify some entities we might be interested to extract. For instance, we want to extract some synthetic recipe, or we want to extract the application or the characterization methods that have been used and describe our images. So, yeah, this is actually the three main, EXSCLAIM has three main components, which is actually the journal scrapping or the HTML scrapper, the caption separation and context extraction, and then the figure separation. So this is how the journal scrapping looks like. So by having our query, then it's actually going through the search webpage of the journal, and then we extract all these related HTMLs. So this is how a paper looks like in a journal, right? So it's a full image and with all these smaller subfigures. And a full caption that describes each of the subcaptions describe each of the subfigures. So we want ideally to be able to extract all the captions and separate all the subcaptions and know, for instance, that the subcaption C is associated to subfigure C. So this is what the caption separator is doing. And then the third part is we separate the figures actually. So at the end, we end up having a full data set with all the smaller images, so each individual image, and then each individual description for each images. And we save all this information in the JSON files. So yeah, so for instance, we have this large image, and then we want to, we use this figure separator component, and then we just split all these images to the smaller ones. So how this works is actually behind that is a vision model. So that was trained based on, actually, we used the crowdsourcing platform from Amazon. So, and several MTurk workers, they were asked, so by giving these large images, they were asked to draw boxes around these images. So this created a large data set of more than 3,000 annotated figures from the users that work in the Amazon Mechanical Turk. And then the visual model was trained so that was able to separate this large figure to all these subcaptions. So this is the figure separator component. And also, we are able to extract some, if an image is a master image or if it is an inset image. So the classifier is able to classify if it's a master or inset. And this information is also given in the JSON files. So yeah, this is big picture. Having EXSCLAIM, we have a large image, and then we just be able to separate all the images, and then having an image caption datasets from our search. So the other part is that, okay, now I have image and text. So how can I use them? What can I do with that? So there are several models that have been developed, mostly using like BERT. So they have used like BERT models that were trained in the whole Wikipedia literature, let's say, and then they retrained them with some human annotated labels so that they can extract some entities. So some distinct examples are MathBERT or MathCBERT. So the thing is that the language models should have like material science knowledge to be able to understand materials related tasks. So this was and this is actually how it will look like. So this is a caption that was extracted with EXSCLAIM, right? And we want to extract some entities, which is maybe the application, the characterization method, the sample description, the material, the property, and the synthesis method. So by using the materials BERT models, we can actually, given all of our captions, we can identify the entities we're interested in by applying these models. However, this task has been simplified a lot by now, living in the era of ChatGPT. So for instance, by using ChatGPT, I don't need to train my model, my BERT model. I just need to give one example. So I can give one few short example to my ChatGPT. Let's say this is a caption, and then this is what I want you to do. I want to identify all the entities. And then for all the entities I have in the text, I want to tag them in which of these categories they belong. So entity and tag. And I just give one example to my ChatGPT, and then given any of our captions, we are able to extract all these entities. And actually, yeah, I'll go through the examples, live examples running the code. I just wanted to give an overview how what we're going to see running the code. So yeah, and actually now we're going to try ourselves to do this process I described. So we will start by running the Colab notebook. So we will give our queries, and then we'll scrape images and text, and then we will use the extracted captions. So then recognize some entities, and then see how we save our entities. So I will go to the Colab notebook. So, I mean, I have on the link that I shared with the GitHub. So here are the tutorials I want to go through. So now we're going through the first one, which is EXSCLAIM demo, that I'm going to just demonstrate the basic usage of EXSCLAIM. So first of all, we just clone the repository. This takes some minutes. And then we need to load the pre-trained the pre-trained models, like the vision models that has been pre-trained on the image separation and the image classification. So yeah, I mean, you can either try to run them by yourselves or just follow what I'm doing, and so you can just reuse it and try it anytime at your own pace. So yeah, now the models are loading, and so yeah, I'll go through the JSON file that we are trying to set. So here we can give the name. So this is actually the name of the folder we want to save our data. Then it's like the general family we want to scrape, and then also how many papers we want to scrape from. So if we apply like if we want to do the full process, so it takes a long time to scrape, like let's say 100 papers. So usually I run the jobs on the supercomputer. So I just want to I set my text file, and then I send I want to scrape from 500 papers, and then it runs for usually a day. If I want to scrape all the information and also extract the content from the papers. Otherwise, it's just take like less than an hour for 500 papers. So here is our queries. So we want to give like the term. So maybe let's say nano, and then or maybe we add some synonyms. So if it's open source, and then also we want to see in which format we want to save them. So I say that the boxes, or if I want to save the CSV, or if I want to save like the full cropped images. And then we can describe if we want to print the information during the scraping. So here is the one I prepared. So I want to scrape from Nature. I want to scrape only 3 papers, and then I want to search for nanoparticles and silver nanoparticles. And here I give all the synonyms from the nanoparticles. Sorry, we have the file type of the papers. So it's currently you don't need any file type. You just scrape from the web page of Nature. So all you have to do is just to give the journal family name. So Nature, and then it directly goes where it go. I mean, it directly goes to the Nature. Let me actually. So it's to the Nature search. Nature search. So yeah, actually, no, by giving Nature, you actually go to the search here. And then it's actually you search for ag nanoparticles, search. So it automatically gets you to this web page. And then you scrape all the open access articles by the HTML. So they are saved as HTMLs. So now I've set my JSON. We currently can support Nature, ACS, and RSC. Although because there is different, so Nature has different web pages than RSC. So Nature is like RSC and ACS. They use like JavaScript for their web pages. So it can make you need to connect with a driver to be able to scrape from there. So with Selenium driver to be able to scrape from there. So I'll show some examples. So here we just import EXSCLAIM, and then we just run our pipeline. Sorry, the testation. Oh, and if I test the testation, and then the process is starting now. So this takes like only a few minutes because I'm only extracting like the images and the captions. So I don't screen through the whole paper, because the other functionality we added currently is that by getting like a figure, we also search through the main text for the context where the main text is describing this figure. So we can also extract this context and then extract keywords from this context. But the simplest way to run it is just to extract the images and the captions. So now it's running first the journal scrapper. So it goes through the three Nature papers that are related to silver nanoparticles. And then it's just saving the HTML. So when we do this run, we can see that here on the EXSCLAIM folder, we have output, and then it's actually the Nature nano test. So this is the name of the folder I gave. So we'll see that slowly we'll have like the figures, first of all. So it's kind of the figures it found, 1, 2, and 3, larger figures. And then we extract the captions. So yeah, we scrape from more. So yeah, from 3 papers, we have all the images scraped from all the papers, all the 3 papers from Nature. And then we get the captions, we save the captions, and we parse the captions from all these images. And then at the end, the vision model is just cropping all these images and then saving them in the boxes. So this will take some minutes. Yeah, the good thing with Nature, when I scrape from Nature, is that all the images are also very high quality, so very good quality. Whereas if you just scrape from some other journals, the quality of images might be kind of a little bit foggy. Yeah, so for now, yeah, we have finished with all the figures. And then here is the HTML folder is like the 3 HTML files that were detected from the search and that we are scrapping the images for. And then here is kind of a smaller list. Okay, the 3 articles, the captions that we scraped, and then the names of the figures. And then, of course, we have like the exclamation where all the information is here, like the title of the papers, the title of the papers, the URLs, the figure names, the full captions, and then when this run is finishing, then the exclaim is going to be updated with all the geometry as well. So the bounding boxes around these images. So here we can see that we have extracted also like the category. So if it's semantic so what the label of the subfigure and also like the description of each of each of the subfigures and also the category of the subfigures. So yeah, when this search is finished, then the success message will be printed. So that means that we finished with our run and we have all the information like saved here. So all the images, all the figures and all the boxes. So, and I can show actually how if you want to visualize the results, let's say we check into the folder and then we just see the figures. Like here on the figures are this saved. So we can select which one we want to. I mean, I just randomly like select it. I just randomly select some figures so we can see them better here. All the related images. And then if you want, let's say, to just plot subfigure B from a certain image, then we just plot like by the oops, maybe there is only one image here. Also, this one image, so there is no label. So if we have like some images that have like labels, so here it's A and B. So here we can actually see the A part, the subfigure A, and then subfigure B. So when we have, this is what is derived by applying the bounding boxes in our larger image. So from this image, we can we split them to A and B. So, and actually, yeah, I'm going to visualize the data set. So how the information is saved. So yeah, we can also, by reading the JSON, we can have all the information here, the name of the image, the title, the URL, and the name, and the captions. And so now, having all the images from nature and my caption and the descriptions of them, I want actually ideally to be able to recognize some things that might be interesting for my research and highlight or identify these entities on the text. So, and previously, as I said, this was be done with BERT models that were like math BERT models that needed like so much time of human annotation because the human annotators had to go word by word in a paragraph and then annotate if any of this word can be categorized or can be given any named entity. But here with ChatGPT, so you actually need a ChatGPT API code to be able to run this. So here actually this is how the prompt looks like. So I wanted to give the example. So one short example. So I gave 2 captions, and then also I identified which entities I wanted to recognize, and then it's just the visualization. So here we can see that by given like a random paragraph, it identified all these entities, and if we can just highlight them. So if we can see, so it identified like nanoparticle or mirror, which is like the blue, which is like the sample description. So in green, we have self-assembly monolayer. So self-assembly is like the synthesis method. Monolayer is actually some description. So it's doing pretty good job than without even need to be trained. Just by giving like 2 examples, it's able to understand all the entities we have in our text without much fine-tuning or without need for training. So yeah, this is the simplest example how we can run EXSCLAIM and how we can use the information. So if someone has any question like on this notebook, otherwise I can proceed to the second notebook. Okay, so this is based on nature papers. So now we also have the possibility to run some HTML files. Because, for instance, for my research, oh, that's not good. So yeah, for instance, for my research, I don't want to scrape like all the Nature papers because I don't know how reliable the synthetic methods are. So I was only wanting to scrape from a smaller subset of papers of a certain group that I know that they do the synthesis. I'm sorry, you have some question? Yes, yes. So yeah, we support ACS, Wiley, and RSC currently. So the thing is that because ACS and while in RC they use like a JavaScript methods, then you need to scrape from them. You need to connect like your Google driver. So this is kind of the code here. So I'm not going to connect it now. I'm just going to scrape from Nature because it takes less time. But if you want to scrape from RC, ACS, or Wiley, you need to connect your Google driver and then to make sure that your driver is connected because we use the Selenium stealth, so Selenium WebDriver services to scrape from this dynamic JavaScript web pages. So for now, we just need to install like the LangChain, the OpenAI versions, and then some tools from transformers. And then it's advisable to restart the time. So I'll also create like a folder here. So where I'm going to add my HTML files. So HTML files. And I will upload like a file that I've been working on. So for my desktop. So it's HTML. Yeah. It's from some certain type of polymers we're interested in to extract the information. So I just downloaded all the certain HTMLs that I wanted to work on. So and I mean EXSCLAIM is mostly working on open source, is working on open source scraping from data. But if you have access to through your university or through Argon account to non open source sources, and you can just download them, save them HTML, and then load them to the HTML folder. So it's also advisable to restart. Okay, and then we start them down, and then we can explain. So yeah, I hope that it will run without GPU. So it might take longer with CPU only, because yeah, GPU is always better. But yeah, I'll try this. And I'd also start installing the tools from the other notebook. So EXSCLAIM is installed. So again, we import the pre-trained models. And then here, the query is the same, but this time I just give, I don't need to give like a general name or a certain like query because I know what I'm looking for. I have downloaded already my paper that is related to polymers, and I just want to give the folder name where I have saved my information, and then also my OpenAI key, and then also the save format. So if it's like boxes or subfigures. So and again here we can also select which tools we want to use. So if you want to do a figure separation, caption distribution, so journal scrapper is false because we don't really scrape from journals, but HTML scrapper is true because we're scraping from a certain like HTML page. So this might be slower this time because it's on CPU only, but then when it finishes, we will be hopefully able to see like again a folder inside EXSCLAIM output, where it's the it's HTML Reynolds. So I need to test this one. So yeah, the file is named HTML. Yeah, we see now the HTML Reynolds, and then we see that the figures have been extracted. So yeah, actually, this is the figures. This is the HTML, and then this paper has like 4 figures, so 4 larger figures. And then now we're just scrapping, assigning all the sub captions to each of the sub figures, and then also separating the figures. So this will also take some minutes. And you can it can support so it can support any type of HTML you upload. So if it's this is kind of a nature HTML, so you can support RC Wiley ACS as well. So any type of HTML, only if you just have RC or ACS, you need to install as well the driver. And then here I have driver is unknown. So then you'll just set driver equals driver, and it will be able to extract information from ACS or the other types of journals. So yeah, here we have extracted even more information. So we have extracted the caption from these images. So we can see the process that is saved here. And then it keeps extracting the captions, and then now it's actually running the figure separator. So from the larger figures, it will just go to the split them to the subfigures. So we will be able to see them hopefully in a bit in the image folder. Yeah, if you have any questions or anything that might be useful for you, I'm happy to answer.
So how this will work with PDFs, for example? Is it possible, or HTML is the best format?
So currently, it's HTML, the best format, because the way they are saved, the data are saved. But actually, yeah, I mean, I've seen currently that GPT can pretty well handle PDFs now. So I think the next thing I'll try to incorporate is to have PDF support as well, because currently it's just HTML support. So you upload your HTML files. So yeah, now we see the run is successful, and then we can see like the images. So we had 4 images, and then for each larger image, we have the extracted like all the subfigures. So it was split A, B, and C. Yeah. So this was by using the HTMLs. So yeah, I know PDFs are more useful to scrape from. It's easier, but currently the format that the HTML is, because we use actually the libraries Beautiful Soup and Selenium. So they actually work for if you have defined the on the HTML format to understand, okay, what is an image, what is a caption, what is the main text. So yeah, we need to, if you have a PDF, we need to convert it first to HTML format and then scrape from that. Okay, so that was the 2 examples, and then I'll go through some actually how we can use this data now. So yeah, I just show you how we can either search from Nature web pages or from any other general web pages to get image and text data, or by using our own HTML files to get our image and text data. So then an example that I found very interesting is like how to use like clip model. So that given the image text pairs. So what this clip is doing is based on contrastive learning, and it is trained jointly on images and text. So the images are encoded, the text is encoded, and then images that can be described from this text, they should found close to the same space at the training. So this clip model has been trained with billions of image captions pairs, not only dogs and cats, but more even scientific images, and a very diverse data set of images and texts. So by having this clip model, it's called zero shot. So that means that we don't really need to fine tune it most of the cases, and can pretty good identify it can be pretty good applied to our types of images. So, for instance, I have extracted like lots of images from EXSCLAIM, and I can categorize them if they are so like mostly like films, if they show like molecular structure, or if they show absorption spectra or X-ray diffractions. So for my project, actually, I was able, by projecting my data sets in image and text, by using CLIP, I can, because I'm only interested to extract the absorption spectra. By using CLIP, we can query it, and then just from a whole bunch of images, we can only understand which of them are related to our interest, and then just get them, get actually only the absorption spectra out. And actually, I have an example how this can work. So this is my third notebook. It's about how we can use CLIP for our data set. So I have actually uploaded already the folder. So it's actually from the EXSCLAIM JSON. So after we finish the EXSCLAIM JSON run, so here I have one because it takes a lot of time. So I just I will upload one I have ready already. So it's this one. So it takes some time to install again CLIP this time, and the OpenAI to this notebook as well. And then it's actually, I'm doing this process. I'm having my data set. So my experimentation, I have all my, in a folder, all my images. So for instance, I have this folder, but for this time, it's for microscopy images, the data set I provided an example. So here we have the output. So imagine that we have this folder with all the images. So all the extracted images, maybe all the images, or all the full figures. And then it's actually the exclamation that has all this information. So this is the whole information that I have on the exclamation. So it's actually the boundary boxes. So how the subfigures look like. Oops. So the so the bounding boxes and the category. The caption, and then also the context. So using ChatGPT, I also extract the context from the main text that is related to the main image. I have the caption of the image, and I also have some keywords that might describe this image. So let me see if it's still running. So yeah, the data are already prepared. So just have the code how we can create a CSV file with all the information that we run. But actually currently I have this CSV file already and the images. And we just using the CLIP model, we can actually import CLIP. So CLIP is has the pre-trained vision and text encoders. So we can select which model we want to use from CLIP, load them. And then we get the text embeddings and also the image embeddings for all our images. So this is kind of what we expect to see at the end. So if we run successfully this notebook, then we have a data set by having like all the caption summary that I extracted the tags with GPT, the image, and then the image embedding and the text embedding that is related to each of the images. And then actually, when we want to query the images, so we can actually, the tests I was running is that I have a whole bunch of embeddings of text and image, and I just want to have a query related to graphene, for instance. So when I query graphene, then we will see that it will identify from my data set those images that are mostly related to graphene, and then this can be proven by the tags. So actually the tags is graphene. So it's not trained on tags, it's actually trained on all the full captions and the images. So full captions and images, and then if I query for a certain thing, or I describe around graphene crystal structure, for instance, and then this is actually the top 4 images that can be shown to me. So and also the similarity score based on the contrastive, based on the cosine similarity that is measured. So I can query a large data set and just keep only those images that are related to my research or that are of my interest. So yeah, actually this is the code. Yeah, if we run it, so I don't want to run out of time. So this is how it will look like. And we can query it with any other type of information we want. And so yeah, so very quickly, I want to go to the last part to explain a little bit about what is plot to spectra and how it works. So plot to spectra is another tool, which is very relevant to EXSCLAIM, because EXSCLAIM gives us the information, image and text. Okay, we have clip that we can use it to train it with both text and image and query our datasets. We can also use ChatGPT or name recognition to use the text that we extract. But we also, the other tool is plot to spectra, that we given some images that are related, for instance, to spectroscopy or Raman spectra, Xamen spectra, we can actually digitalize them. So and create data sets for material science. So for instance, this is what we get from the literature. Then we want ideally to be able to learn all the XY coordinates for all of its lines, and to split them and separate them. So currently there is the most famous tool is WebPlot Digitalizer. So you can upload an image, for instance, and then you can ask it to separate it based on the color, or you need to click one by one all the points to identify that you want to extract the coordinates of this line. But it's kind of not sustainable because it's so time consuming, especially if you have lots of images. So with plot to spectra is a tool that is actually two components. So it's an axis alignment module. So by giving the image, we can identify where are the axes shown, and then to know like the peak distances. And then the second part is how we do like the data extraction module. So by using like the CNN segmentation model, so we actually classify the foreground pixels with the background pixels, and then we keep only those information that is related to plots. And then we apply optical flow technique to understand which of them are the different plots. So actually, as an example, this is how the axis alignment module looks like. So by giving an image, we identify which are the tags of our axis, and then we extract these numbers. So we know what is the step of here. And then the data extraction module, which actually we use the CNN-based model that was fine-tuned with specific spectral images. So for instance, we actually separate the pixels. So if a pixel is relevant, so if a pixel belongs to one of our lines, is classified as is classified as as the pixel we want to capture. If not, if it's on the background, then we just it's not relevant information, so we split it from the good pixels. So in that way, we remove all the noise and all the irrelevant information from the images. So we classify them as background and foreground. And we map the pixels to feature vectors, and then we can cluster them so that we know in which instance they belong. So as an example, actually here, we have this plot. So the information about the names of the plots is not relevant. So we only care about, or the axis numbers is not relevant. We only care about these lines. And then how the optical workflow works is that if we pick a point here at T0, so then we compute the flow in each point, and then we can estimate the position at point T0 plus dt. So we do that, and then we actually track each of the points. So we just track to find the neighboring points. So and that way you can identify that all these pixels, for instance, belong to this line. All these red pixels belong to the red line. And that's how you track the pixels, and then you can just separate and understand that you have 3 different spectra here. So because I think we ran out of time, I also just the notebook. So just very quickly. So I have it actually here because my Colab is out of GPU. So here is actually, we need to install the versions. So, and actually there is kind of a little bit of trouble with the vision models that are used. So it should be on the very specific versions that were trained on. So the MMDET should be like 1.34, and MMCV should be 2.11 to be able to use the pre-trained model. Then we can select our options. So where are our checkpoint files, the pre-trained model, where are our checkpoints for the axis alignment, the pre-trained models. So there are several like tunable parameters. So how well we want to identify the lines. And by setting that, we can do the alignment. So by running this part, we just do the alignment and identify the axis of our image. So I have the image I have here is the example image. So it's this image. And then we run the first bit of the code and we identify the axis lines, and then we run the second part and we can extract actually the lines separately. So at the end, if we run all this code, we can see that the output of the model has identified each of the lines, so that are 4 differently by tracked by the optical flow, and also all the tags here, all the tick labels here as well. So, and we know that in this starting image, by applying protospectra, axis, and optical flow, we actually separate the initial starting image to this XY coordinates of each of these lines, and then we can save them and have a data set ready for that. So yeah, and with that, I think this is all I wanted to show and say. So if anybody has any questions, or if anybody is interested how to use these tools to their own research, I mean, I'm happy to contact me and we can see how we can apply it. I mean, we can, of course, like repropose them. So for instance, we can add PDF support to EXSCLAIM. So depending on actually on the user case. So I'm mostly using for polymer data. So I'm just reproposing EXSCLAIM or protospectra based on the type of data I have. Because for instance, if we have some spectra that are overlapping, then it's kind of more difficult to use the optical flow technique to separate these lines. So we need to have to do some color clustering and separate it via color. But it depends again what type of images that we have. And so yeah, we just retrain the models or repropose them. So I think that was all for me.